Yeah, good morning friends. Intrapituitary cytokines and the growth factors. They work by autocrine and the paracrine mechanisms, and they affect the pituitary gonadotropin synthesis as well as the release. Today we will learn about the inhibin, activin and the follistatin. Let's start with the inhibin. Inhibin, it's predominantly secreted by the granulosa cells. Activin and inhibin, they are the peptide members of the transforming growth factor beta superfamily. It is made up of two dissimilar peptides which are linked by the disulfide bonds. How does it work? FSH stimulates the release of inhibin, but inhibin suppresses the FSH. Also, it prevents the upregulation of a GnRH receptors. In fact, at high level of inhibin, it actually causes the degradation of the gonadotropins. There are two types of inhibins, inhibin A and inhibin B. The inhibin B, peak level in the mid-follicular phase of the cycle, it actually amplifies FSH withdrawal from the other follicles. So, it allows emergence of the dominant follicle. Once there is a dominant follicle development is there, with the effect of the LH, there is a ovulation, and at that time inhibin A predominates. Now, our next molecule is activin. Our second molecule is activin. It is also a member of transforming growth factor beta superfamily. It is predominantly secreted by the pituitary gonadotropes as well as the granulosa cells. As the name suggests, it promotes the secretion of the FSH. It inhibits the release of the prolactin, ACTH and the growth hormone. And it also augments the pituitary response to the GnRH by enhancing GnRH receptors. Inhibin and the follistatin, they inhibit the effects of activin. Our third molecule is follistatin. The third molecule is a follistatin. It is a glycopeptide which is secreted by the variety of the pituitary cells including the gonadotropes. Interestingly, it is a FSH suppressing protein and it inhibits the FSH synthesis, FSH secretion and FSH response to the GnRH. Activin stimulates the follistatin and inhibin inhibits the follistatin. So, how does endogenous opioids control the GnRH? Endogenous opioids are the endorphin, enkephalins and the dynorphins. Endorphin, which is mainly synthesized in the hypothalamus, it has an alpha, beta and the gamma subunit. The precursor molecule is a pro-opioid melanocortin and it has an action which is similar to morphine. Endorphin inhibits the GnRH release. This is the main mechanism of action. It prevents gonadotropin secretion, but it has no effect on a pituitary response to the GnRH. Ovarian sex steroids positively affect the endorphin and this endorphin inhibits GnRH. Beta endorphin, it is a 5 to 10 times more potent than the morphine. Next opioid is enkephalins. Basically, it controls the autonomic nervous system. It is a pro-enkephalin A and it has a methionine and the leucine enkephalin. Dynorphin, it is a precursor molecule pro-dynorphin. The action is similar to endorphin. Basically, it is related to the behavioral action, but it has a very high analgesic potency. Endorphin basically controls mood, appetite, temperature and the behavior.